Welcome to ASHG 2020. Please listen to this brief review of technical tips and guidelines for ASHG sessions. If you have questions for speakers, please use the question and answers tab, not the chat tab. Moderators will use these and answer as many questions as possible during the Q&A. ASHG encourages attendees from all career stages to ask questions. Respect your colleagues' work, and remember, ASHG's policies prohibit video capture, screen recording, screenshots, audio recording, or taking pictures of speakers and their slides. Use the chat tab to interact with fellow attendees, to just say hello or share your excitement about the content. Stay connected and share your thoughts through our social media channels using hashtag ASHG20. Respect speakers' preference if they opt out and ask you not to share or discuss. Speakers will note this at the beginning of their presentation. Check out the Handouts tab to download additional resources if provided by presenters. Thanks for your attention and enjoy the session.
Hello, I'm Tony Winshaw-Boris, this year's ASHG president. Welcome to the Presidential Symposium, the big tent of genetics and genomics, research, translation, and advocacy. I will briefly introduce our distinguished participants to allow as much time as possible for their presentations and discussion. My co-moderator, Dr. Eric Green, is the director of the National Human Genome Research Institute and a physician scientist studying genetics and genomics. Dr. Green has led NHGRI for many years and has been a major ASHG partner in leading the Institute's research and educational programs as well as other initiatives. The panelist speakers are Dr. Gary Gibbons, who is director of National Heart, Lung, and Blood Institute. He is a physician scientist leading the third largest institute at NIH. Among his own research accomplishments, Dr. Gibbons made major discoveries related to the cardiovascular health of minority populations, vascular biology, and the pathogenesis of vascular diseases. Dr. Diana Bianchi is director of the Eunice Kennedy Shriver National Institute of Child Health and Human Development. She is a physician scientist and has been a member of ASHG for more than 25 years. Dr. Bianchi's research is focused on prenatal genomics, particularly non-invasive prenatal DNA screening, and investigating the fetal transcriptome to develop new therapies for genetic disorders. Dr. George Yancopoulos is president and chief scientific officer of Regeneron. Under his leadership, Regeneron is focused on addressing a broad range of serious medical conditions, including eye diseases, cancer, rheumatoid arthritis, asthma, atopic dermatitis, pain, and infectious diseases, including, of course, SARS-CoV-2. Mary Woolley has been president of Research America since 1990 and has been a tireless advocate and strong supporter of science. Research America is the nation's largest not-for-profit membership-supported grassroots public education and advocacy organization and is committed to making medical and health research a higher national priority than it already is. So let's begin, please. Dr. Gibbons, welcome.
Well, thank you very much. If I could have my first slide. It's really a pleasure to join in this panel. As you say, to come under the big tent, part of the circle of partners that are necessary to fulfill the mission of the NHGRI. I have no disclosures. And so I want to put forward both a challenge and an opportunity under that big tent, in which we'd like to invite various elements of the ecosystem that's gathered here to address complex problems. The one I'd put forward for you is one that we think poses an opportunity to reduce health disparities, with a particular focus in this particular presentation on women at high risk of cardiovascular disease and maternal morbidity and mortality. This is germane because it reflects a collaborative partnership that I have with the subsequent speaker, Dr. Bianchi, in child health, in which there's a convergence of risk factors that promote adverse pregnancy outcomes, as well as set a trajectory for increased risk of cardiovascular deleterious outcomes. The map on the left gives you a sense of the maternal mortality in the United States. And to this expert group of geneticists and genomicists, I think you can see in that geographic pattern that this bespeaks not only potential genetic biological determinants, but a major signal for environmental influences and particularly the social determinants of health that in a complex dynamic interplay to promote these complex set of problems. In the middle panel, you see the disparities by race ethnicity, in which in particular, in this case, a dramatically high rate of pregnancy-related deaths among African Americans compared to whites in this country. I must say that every time I look at this histogram, I'm appalled at this health inequity. This is far out of proportion to advanced countries around the world. Indeed, there are parts of the world that have lower morbidity and mortality rates than those seen in African American communities. This is a health disparity that, quite frankly, is an embarrassment and something that needs to be addressed. It's relevant to the NHGRI in particular because many of the complications that promote both morbidity and mortality relate to disorders of bleeding, hemorrhage, cardiomyopathy, hypertension, that are obviously within our purview in this context of pregnancy. Moreover, there's an interesting interplay and iterative interrelationship in which risk of cardiovascular disease appears to predispose to adverse pregnancy outcomes. And conversely, individuals who experience adverse pregnancy outcomes tend to be on a higher trajectory for adverse cardiovascular outcomes in a sort of dynamic or spiraling kind of way. And so this is an opportunity for prevention and preemption of disease that can address health disparities over the short term and long term. Next slide, please. In that regard, in many ways, one can conceptualize pregnancy as a sort of stress test. Forgive me for my cardiologist roots, but that's a way of potentially identifying women with a long-term risk trajectory that is revealed by the biological, physiological, systemic stresses of pregnancy that then in many ways foreshadows many or reveals many cardiovascular either events or trajectory of increased risk. This stress test, as you can see, it has an overlap with cardiovascular risk factors that are, again, predictive of future cardiovascular events. And so, again, there's this parallel in which the longstanding cardiovascular risk factors defined by Framingham over a heart study over 40 years ago or more of high blood pressure, diabetes, smoking, an unhealthy diet, obesity, excessive weight gain in pregnancy, all predisposed to these adverse maternal outcomes, but also presage an increased risk of cardiovascular disease over the long term. And to this audience, you are aware of the sort of genetic underpinnings of a variety of those phenotypes singly or in combination. Next slide, please. And indeed, one of the features of that as women age and the age of women who childbear increases and the number of comorbidities increase, the number of these adverse outcomes appears to be increasingly problematic, particularly in the cardiovascular realm. Indeed, hypertension, and in this case, cardiomyopathy are particularly problematic and particularly difficult, challenging in African Americans, where we again see these racial disparities. Clearly, this is a phenomenon of peri and postpartum cardiomyopathy that we're still seeking to understand. But we are encouraged by the insights from molecular genetics and genomics that appear to be converging to give us new insights. And here we cite some of the work of Cricket Seidman and others, in which, in this case, Cricket has begun to identify some of those mutations that are noted in familial cases of heart failure or cardiomyopathy that involve the sarcomeric structural proteins, in this case, titin. If you do sequencing in many of these women with peripartum cardiomyopathy, you start to see variants that indeed appear to be pathologic and potentially predisposing to heart failure and cardiomyopathy. And again, this is revealed in the stress test of pregnancy. And again, suggesting that there is this interplay and trajectory that these individuals are on that is brought out by the stress test. One of the challenges in this regard is, again, the higher prevalence amongst African Americans. But this is where I guess I would make an appeal to this society and this discipline to help us address the inequities in genomic resources that exist that can help further characterize and define what are the functional variants or the pathological variants across various ancestral groups, such that if we pursue the capacity now to sequence the genome, that we'd have an understanding of the potential genetic inputs to these racial disparities that we see in peripartum cardiomyopathy. Indeed, we have a project underway in a collaborative cohort called New Moms to Beat Heart Study, in which we're pursuing that approach. But part of the challenge, again, relates to the skewed genomic resources that exist to really understand the full spectrum of genomic variation in all ancestral groups. Next slide, please. Toward that end, one of the intentions of the NHGRI's effort in this space was through the Trans-Omics for Precision Medicine, or TOPMed program, which very intentionally took an inclusive approach to genomics with an eye toward health disparities to enable data science that would start to chip away at this void within our disciplines, within our fields, within our resources, such that we could address and leverage genomic technologies to gain insight into health disparities. On the left, you can see in this resource that now is up to about 150,000 whole genomes derived from a variety of our community-based cohorts, such as the Framingham Heart Study, Jackson Heart Study, MESA, and the study of Latinos and others. You can see that indeed those of European ancestry are in the minority as we attempt to chip away again and making a more inclusive approach to genetics and genomics. 58% are women in this, and you can see the variety of phenotypes that this covers, obviously skewed a bit toward our mission areas of the NHGRI. And we've placed this in a data resource, cloud-based data resource, in which we're hoping to abide by FAIR principles, findable, accessible, interoperable, reusable, where we hope that there are multiple dimensions of data, not only of the whole genome sequencing, but the imaging data of these longitudinal cohorts, various dimensions of behavioral, social, and environmental elements, all that interact to shape risk of these complex traits and intersecting syndromes. We are layering not only the whole genome sequencing, but measures of the epigenome, transcriptome, metabolome, etc., in addition to the various social and clinical parameters that I mentioned. Next slide. Indeed, during this meeting, there's already been a presentation of TopMed that has described some of the variant calling capabilities, the annotations of up to about 1 billion, I think, variants now in a database. Almost 10 million genomes have now leveraged the TopMed imputation server that takes advantage, obviously, of its ancestral diversity. And so this is a resource in which, again, we invite this community to help us deal with these challenges of addressing health disparities. Next slide, please. Part of our hope is that our circle of partners in this ecosystem will begin to address these disparate trajectories of risk, in which you can see on that left-hand panel, where in particular African American women are on that steeper, earlier trajectory toward either incident hypertension. I could show you a similar one for cardiovascular disease. And you can see that it's starting to infringe on that reproductive life stage. So these are women who are already getting the risk factors at that early stage in life. And so you can imagine what that then does in middle and older age. And we're encouraged by the work of the discipline of this society in using the technologies of genomics and molecular genetics to begin to understand these complex traits in ways that you can start to understand the polygenic, cumulative, and interactive inputs, perhaps of multiple genes and pathways and perhaps networks that are driving these trajectories, and hopefully giving us insight that is more refined than our current clinical risk factors that Framingham has established for the last 40 years, as well as identify new molecular targets that may be amenable to therapeutic intervention, similar to what I'm sure our colleagues at Regeneron may speak to. Next slide, please. So this is an opportunity for precision medicine as we get a greater and more refined picture of the molecular basis of these complex traits. We also hope that this is a means or a lens or an entree into targeted precision interventions and precision prevention, in which we seek to change this trajectory, assisted by better identification of those at highest genetic risk, as well as other clinical variables. And we have suggestions from these observational studies that those, for example, who adopt a healthy lifestyle may modify outcomes relative to their genetic risk alone. And clearly in our other parts of our portfolio, we're designing interventions, indeed dietary interventions of the DASH diet, which we know can lower blood pressure, and doing interventions to enhance the uptake of that dietary behavior. Indeed, we're understanding now its influence on the transcriptome and metabolome. And so we're understanding the molecular basis by which a diet rich in fruits and vegetables and whole grains is able to lower blood pressure. And that in and of itself may give us important new molecular insights and potential targets. Next slide, please. So we're hopeful as I close here with this what if that I hope is something of a challenge and an opportunity to this entire ecosystem of genetics and genomics, health disparities researchers across the spectrum, our policymakers, our private sector, public sector, as we take on complex, vexing problems like health disparities. And I don't need to tell this audience in the context of a virtual meeting in a historic pandemic where these health inequities have been laid bare in dramatic and devastating fashion amongst some of these same high-risk groups of women and men with hypertension and obesity, that this is something that our country must address. We can no longer have the highest rates of morbidity and mortality amongst women who get pregnant. Pregnancy should be a benign event, certainly in communities of color. And so we're hopeful that you'll join with us and bring your expertise to help us define and develop better strategies to do that. So we might help this hypothetical case of Camilla, 35-year-old African-American woman who in her first pregnancy had preeclampsia and mild heart failure. And on her second pregnancy had excessive weight gain and was told at her postpartum visit that she'd had excessive weight gain, was at risk for prediabetes and hypertension, and then discharged from care. How can we change that trajectory? And we'd love your support, your insights, your expertise to help us tackle this problem. Thank you for your attention. We look forward to your collaborative partnership.
Thank you very much, Dr. Gibbons. Next, we'll hear from Dr. Diana Bianchi, Director of National Institute of Child Health and Human Development. Diana?
Thank you, Tony. I've titled my talk, "The Big Tent Can Get Bigger," and I'll explain in a moment. One of the questions that we were asked in preparing this session was, how can ASHG partner with both federal agencies as well as foundations and industry to make the tent bigger? And Tony and I think, are the only medical geneticists on this panel. And this question reminded me of some of the discussions that we've had over the years as to whether there's a need for a separate medical genetics and genomics specialty, or should everyone be a geneticist or a genomicist? And I think the answer to those questions is you really do need both. And in terms of a big tent, from the federal side, one of the questions that's been asked over the years by Eric is, should NHGRI be the exclusive NIH home of genetics and genomics research? And I think the answer to that is no. Does every institute need to be current in genetics and genomics to support a wide community of researchers? And the answer is yes. So in the next 10 minutes, I'm going to give you some examples of what we're doing at NICHD to partner with everyone to bring resources to you, as well as partner with researchers who may not necessarily be in our typical areas of research. Just about a year ago, we released NICHD's first strategic plan in 20 years. And we had a large outreach to external and internal groups. We started with something like 270 suggested scientific themes and honed it down to these 5. And you can see by their titles that all of them have something to do with genetics and genomics. Certainly understanding the molecular, cellular, and structural basis of development, promoting gynecologic, andrologic, and reproductive health, setting the foundation for healthy pregnancies and lifelong wellness, as Dr. Gibbons mentioned, improving child and adolescent health and the transition to adulthood, and advancing safe and effective therapeutics and devices for pregnant women, lactating women, children, and people with physical and intellectual disabilities. So there's something in there for everyone. Well, with regard to developmental biology, one of the things that people often ask me when I speak is, do you collaborate with other institutes at NIH? And we have an enormous record of trans-NIH collaboration. Certainly COVID has increased that, but even prior to COVID, all of the institutes collaborate with each other. So one example is this developmental genotype tissue expression, the DGTEX project, but GTEX has not focused on developmental tissue. So in partnership with NHGRI, we are hoping to establish a resource database and an associated tissue bank to study gene expression patterns in multiple reference tissues during different stages of human development, including both the placenta and neonates through adolescence, building upon the completed GTEx project. So if you're interested, if you have resources and you're interested in applying for that opportunity, the link is below. But there's a major gap in information about developing tissues in children and in the placenta. The INCLUDE project, which is an acronym for Investigation of Co-occurring Conditions Across the Lifespan to Understand Down Syndrome. This is the result of significant increase in congressional support to study Down syndrome. And actually, Dr. Gibbons and I are co-chairs of the steering committee that oversees this major project. And over the last couple of years, we have had a significant effect on the type and the amount of research that's being done in Down syndrome. There are three components to this research. One is what we call the transformational R01s, which are to conduct targeted, high-risk, high-reward basic science studies using human cell models, organoids, animal models, all sorts of preclinical studies. There's been a history of exclusion of people with Down syndrome from clinical research. So another goal of this project is to assemble a large study population of individuals with Down syndrome across the lifespan, and then to include individuals who have Down syndrome in existing and future clinical trials with the twin aims of learning about what can help people who have Down syndrome, as well as learning about conditions that are more common in people with Down syndrome, such as Alzheimer's disease, but that knowledge can also help people who do not have Down syndrome. Another example of a publicly available resource is the Gabriella Miller Kids First Pediatric Research Program, which was established in 2015 to alleviate suffering from childhood cancer. Gabriella Miller died at 11 years of age, all too soon, from cancer, and also to alleviate suffering from structural birth defects by having collaborative research to uncover the etiology of these diseases and supporting data sharing within the pediatric research community. And what this program does is it supports sequencing of cohorts of various birth defects. There's a data resource, which I'll show you in a moment, which is a platform that empowers collaborative pediatric research. It allows you to query, search, discover, and build and visualize synthetic cohorts. It's very user-friendly, and it really is a model of clinical data in a FHIR-based data services for interoperability and coordination. It allows data visualization tools, and you can pull data from multiple sources into one workspace. And we've already seen a tremendous impact just in the last 5 years in terms of these cohorts that have been sequenced. We've been able to provide with this funding 37,000 whole genome sequences from 15,000 different cases. The light blue conditions are the released data sets. They're publicly available. And our team has already responded to over 150 data access requests. Just to give an example, right at the top of the list is congenital diaphragmatic hernia. An enormous number of genes responsible for this condition have already been identified. And you can see that there are both birth defects as well as childhood cancers that are in the light blue. And the conditions that are in black are in various stages of getting ready to be publicly released. It's a tremendous resource. I think in the interest of time, I'm not going to go through the use case, but just to show you that anybody can register, can agree to the terms, and then you can ask your questions. So just as an example, if you want to find out, does anybody with congenital heart disease have neuroblastoma, or are there genes that would predispose to both of these conditions? You make the query, and then you build your synthetic cohort. And then the system pulls in the data and then you can analyze it and visualize it. So it is, I've seen it. I haven't used it myself, but I've seen it demonstrated, and it really is quite user-friendly. We were asked to address health disparities, and we were asked to address COVID-19. These are all challenges in a 10-minute talk. But I did want to communicate with you that NICHD is very committed to addressing COVID-19 in our populations, so children, pregnant women, people with physical and intellectual disabilities. And we are in the process of selecting applications now for a new program called Prevail Kids, which is predicting viral-associated inflammatory disease severity in children with laboratory diagnostics and artificial intelligence. And this program has been developed to figure out which children will go from having either an asymptomatic case of COVID-19 or mild symptoms, and who will go on to have this multi-system inflammatory syndrome in children, which can be associated with life-threatening illness. This MISC, it occurs about a month after the initial infection with COVID. And by the way, children are increasing in terms of the percentage of all COVID cases. Currently, they're around 11% of all of the COVID cases in the United States. So we are working with pediatricians around the country to enroll a diverse cohort of children who are SARS-CoV-2 positive. We are aiming to recruit a diverse population with predominantly Latinx and African American children, because they are responsible for about 60 to 70% of the cases of COVID-19 in children. We'll be collecting their data and biospecimens prospectively, and then we will be retrospectively doing genomic studies, analyzing the immune systems and immune genes to develop an AI machine learning predictive model to be able to understand which children are specifically at risk, not only for the MISC, but also potentially for long-haul symptoms. The review actually is going on today and tomorrow, and we will be making these awards by the middle of December. So in summary, in this very brief presentation, genetics and genomics are woven throughout NICHD's strategic plan. We don't just focus on children. We actually fund about 50% of NIH's portfolio on maternal health. Effective genomic efforts employ team science. We work across NIH institutes and centers on many initiatives. I've given you a few examples of this. And NIH and NICHD support many resources that are freely available to the community of researchers who are watching this video cast. And we really encourage you to explore and to identify causes of diseases related to human development and reproduction. So I think that the tent is already big, but it can certainly get even bigger. So thank you very much.
Well, thank you, Dr. Bianchi, for that great talk. Next, we'll have Dr. George Yancopoulos, President and CEO of Regeneron. Dr. Yancopoulos.
Thank you so much, and I greatly appreciate the invitation to be part of this panel. Before I get into our views and efforts as to how human genetics can support COVID-19 pandemic efforts, I've been asked to provide a short overview of our ongoing therapeutic efforts on COVID-19. Many of you may be aware that just last night, we released overwhelmingly positive results from our confirmatory clinical trial in COVID-19 outpatients testing our anti-COVID-19 antibody cocktail. To provide some background, we were only able to rapidly create and develop our antibody cocktail because of decades of investment we had made in developing innovative leading technologies in the antibody space. And our cocktail consists of two potent neutralizing fully human antibodies targeting distinct parts of the critical receptor binding domain of the SARS-CoV-2 spike protein. We previously utilized an analogous approach to develop a similar cocktail to target the Ebola virus. And just last week, our Ebola antibody cocktail became the first treatment of any kind to be approved by the FDA for Ebola, based on providing a significant mortality benefit in both early and advanced Ebola patients, as published in the New England Journal of Medicine. Obviously, our success with Ebola created a lot of hope for our cocktail approach in COVID-19. The use of multiple antibodies in our antibody cocktail has been shown in preclinical experiments to prevent the mutational escape seen with single antibody treatments. And that's why we chose to go with this approach in human trials, out of concern that single antibody treatments could seed viral escape mutants into the broader population, as was seen with single drug approaches in the early days of HIV, and that such escape mutants might then be more resistant both to other antibody treatments and possibly even to vaccines. Before going into human studies, the antiviral activity of our antibody cocktail was validated in non-human primate studies, showing viral load reductions in both the prophylactic and therapeutic settings. Our cocktail is now in multiple human trials evaluating distinct stages of the disease as a prophylactic treatment in people at high risk of contracting COVID-19, in recently infected outpatients to prevent progression to more serious disease and hospitalization, as well as in advanced hospitalized patients. A few weeks ago, we reported initial descriptive results of our cocktail in 275 mild to moderate outpatients, showing potent antiviral activity and also associated clinical benefit of reducing need for further medical attention. One of the most important additional learnings from our early study related to the natural history of infection, which we learned from studying placebo patients in the trial who we didn't treat. What we saw was that many placebo patients rapidly make their own endogenous antibodies to the virus. And these people rapidly reduce their viral load on their own and do very well without requiring further medical attention and hospitalization. However, we saw other people more slowly make their own antibodies and then more slowly clear the virus. And these individuals were at much higher risk for requiring further medical attention and hospitalization. As you might expect, what we saw in our early analysis, looking at the treatment groups, was that the antiviral benefit, as well as the reduction in risk by giving our cocktail, was largely driven by its effect in these slow immune responders. That is, our cocktail seemed to convert the high-risk slow immune responders into the equivalent of fast immune responders who did well. Just yesterday, we released prospective and overwhelmingly positive results in a larger analysis involving 524 additional early outpatients, confirming the potent antiviral activity, as well as demonstrating a statistically significant ability to prevent the need for further medical attention, such as hospitalizations and ER visits. And once again, and most importantly, showing that these benefits of the cocktail were most pronounced in the highest risk patients who were slow to mount their own immune responses. Obviously, these are exciting data, and we are in the process of discussing emergency use authorization for the cocktail with the FDA. There are parallel efforts from Lilly on a similar antibody approach, which also looks promising. And as I'm sure you all know, many others are working on a variety of vaccine approaches. Obviously, we need multiple successes with all of these approaches because the world desperately needs multiple weapons to fight back against this horrific pandemic. Now, switching gears to our efforts on the genetics of COVID-19. As I am sure many of you know, our Regeneron Genetics Center, led by Aris Barris and his senior team of John Overton, Jeff Reed, Gonzalo Becasis, Manuel Ferreira, and others, is a uniquely integrated research initiative that has taken on one of the world's most ambitious human sequencing and analytics efforts using state-of-the-art automation and cloud-based informatics, and involving over 90 critical collaborations all over the world, from Geisinger Health Systems to the UK Biobank and to many others. And our effort has already sequenced nearly 1.5 million individuals, almost all linked to detailed electronic health records, with the goal of making genetic and disease associations that can both identify drug targets and aid in drug development. For COVID-19, in addition to the above initiative and collaborations, we joined the COVID-19 Host Genetics Initiative led by Mark Daly and Andreas Agana and their team, and also made an important alliance with Ancestry.com to further increase the power of our approaches for this pandemic. We also made a publicly available browser that allows anyone to search our collaborative UK Biobank data exome variant associations with COVID phenotypes. We've already been learning a lot about possible genetic associations, and for example, learning the value of much larger data sets and more detailed phenotyping. For example, we have found that certain genes thought to be generally associated with COVID-19 might only be associated with specific stages of this disease. And we found that larger data sets often contradict associations described in early high-profile publications using smaller populations. And we also learned the hard way that while genetics can be very valuable, this is only true if you have the information in time. In the very early stages of the pandemic, there was a lot of excitement about the role of excess inflammation driven by interleukin-6, based on small studies in China and Italy using interleukin-6 receptor blocking antibodies, which rapidly became the standard of care around the world. As we had one of the two FDA-approved anti-IL-6 receptor blocking antibodies, we felt the urgency to perform a large, well-controlled study to confirm the value of these therapeutics. A huge effort involving a $100 million investment, but most importantly, also putting so many sick patients at potential risk. After all of this effort, only to find that IL-6 blockers show little benefit, but luckily no harm. Roche subsequently confirmed our finding using their own IL-6 blocker. Months later, using the above large collaborative genetics resources we have been developing specifically for COVID-19, we generated definitive genetics data showing that IL-6 loss of function variants, which clearly are protective in known IL-6 driven diseases such as rheumatoid arthritis, have no effect on COVID-19 infection or hospitalization or severity rates. Many of these findings were presented earlier at this meeting, yesterday, by Jack Kosmicki, who led this work along with Julie Horowitz in our group. So in this case, genetics confirmed our large definitive clinical trial, showing no benefit of targeting the IL-6 pathway. Though obviously, we'd have much preferred it to be the other way around, and that the genetics had been first, and that we could have saved so many resources and also avoided so much potential risk to so many people. To me, that is the story, the power of genetics, but the need for us collectively to be creating the large-scale databases and the analytics abilities so as to be able to use the power of genetics to prospectively allow us to make better decisions, to increase our success rates, to waste less resources, and to put less patients at needless risk. Thanks so much for your attention.
Thank you, Dr. Yancopoulos, for that great talk. Next speaker will be Ms. Mary Woolley, the President and CEO of Research America. Ms. Woolley?
To be with you, and I'm going to give a very different kind of talk. I'm going to talk about advocacy, starting with a glimpse of a very stressful year to date, where we need success of the kind that George was just talking about, and that all of you are working toward every day. This isn't news, nothing there will be news to you, but it's the atmosphere in which we're all working, including our work as advocates for your work. And we're advocates for both policies and for resources in both the private and public sectors so that we can find answers sooner rather than later to what ails us. I was really struck by this remark by Don Berwick, the former head of CMS, fairly early on in the pandemic, as talking about what comes next as a series of choices rather than fate that is dealt to us. And choices rightly implies that there's action involved, action by each of us every day, from doing things that are protecting our own health, and that's that of our family, friends, colleagues, and society at large, and doing things to make sure that those who have less access, those who, for example, as Dr. Gibbons was talking about, who are struggling with the outcomes of health disparities that in truth we've known a lot about for a long time, but haven't done anything about. And it's time to overcome that. You do have agency. You can change the image of things to come, as was remarked often by our chair emeritus, former Congressman John Edward Porter, for whom the largest research building at NIH, the Neuroscience Center, is named. This involves being an advocate, getting involved, standing up for what you do and why it's important. Right now, our advocacy includes advocacy for voting, and for voting in a way that will make science strong and keep it strong. There's a lot of interesting data on this website that I encourage you to share with others, particularly those who might not have taken the time yet to vote. So one of the things that we do is commission public opinion surveys, and we do this to keep a finger on the public pulse. There's a lot of good reasons for doing that, not least that understanding public sentiment is a way to make sure that whether you're advocating for policy or resources or anything, that you're going to succeed rather than fail. Because if you're aligned with public attitudes, you have a much higher chance of doing so. Now, the kinds of surveys we commission are don't generate the kinds of data that you're generally familiar with, and yet you see data from surveys like this every day, and in an election season, you see it multiple times every day. So I'll show you some survey data, starting with a couple of items from a survey that we conducted in conjunction with the ASHG about a year ago. It was December 2019, with the results coming out the following month. And I hope you'll see here just how positive people are in the words they use to describe human genetics research. At a time where we hear a lot of negative things about science and lack of trust in science and so forth, it's good to remember that, in fact, people do see science as a solution, as a big part of a better future, a healthier future. Another item from that survey, and I encourage you to look at it, I'm certain that it's available through ASHG, is that Americans think more research is needed in human genetics. When you see public opinion survey data, you should always combine the positives and the negatives and look at them with more attention than the breakdown within the positives and the negatives. The reason for that is it's just understood that there are some people who will never say 10 out of 10 or 1 as the highest or even the lowest. They want to be somewhere closer to the middle. It's their sentiment, whether it's more positive or more negative, that is the thing to watch, especially over time. Now, turning to some more recent survey work, I think it's interesting to see that a very strong majority of Americans across the political ideology landscape, if you will, see that, are inclined to say that the work that scientists do benefits me. Does it benefit me? Yes. We would like to be 100%, including for those people who aren't getting the full benefit right now. But you can see here that there is a very strong preponderance of the population, again, across the political spectrum, with dipping a little bit among independents, and also dipping, and this is discouraging to us, a little bit surprising, dipping for a younger age cohort. The 18 to 29-year-olds here are less likely to say that the work of scientists benefits them personally. We can talk about that if you're interested, but we would be speculating. We don't know the real answers to that. Now, of huge importance, really delivering a mandate to our elected officials right now. 4 out of 5 Americans agree that it's time for an urgent refocus on science, urgent, driven by the pandemic, but not only because of the pandemic. You can see in the next slide that people assign an urgent or high priority to many issues of the day. Of 20 that we inquired about, here you see the top 6. And we also asked people to indicate whether they thought science, technology, and innovation was important to addressing these issues. So number 1, ending COVID-19, is followed at the same level by finding new ways to prevent, treat, and cure illnesses beyond COVID. This, again, is in mandate territory for those who are making decisions about the resources that you and your colleagues need to attack these priorities and make a difference for the American public. Now, the relationship between scientists and their elected officials has been somewhat opaque from time to time and in the minds of many people. And yet the public believes that scientists should take the time to inform elected officials about their research and its impact on society. And I don't mean by sending your latest article to your senator or your congressperson. It's critically important to have that conversation, even if it's virtual or if it's on paper or electronically, to do it in a way that meets the needs of your audience, rather than as though you were talking to fellow scientists. Now, we also know that people say that it's important for elected officials to listen to advice from scientists. Lately, there's been a lot of concern about that. But the American public, actually for years and years and years, in surveys we've commissioned, have indicated how truly important it is that elected officials listen to scientists. Now, I'm glad to report that we've seen a stunning increase in the response to a question we've asked for 25 years now, asking people an open-ended question: can you name a living scientist? And if so, please give me that name. And we saw a big jump up, 20% increase to now 29% of the public, getting close to a third of the public, who say they can name a living scientist. And it's probably no surprise to you that the person who is named is our friend Tony Fauci. I think it is still shocking and really not a good sign that only 29% of the American public can actually name a living scientist, and some of the people they name are dead. We've got some work to do as a community to overcome the invisibility of science. And I'm not going to show you all the rest of the slides that make the case about invisibility, but people don't know where science is conducted either. And they don't know it's conducted in all 50 states. How are we going to overcome this? Well, one way is by making a push, being an advocate for, another way of putting it, for stronger STEM education. Of course, this would make a difference over time. Again, combine the strongly agree and somewhat agree, and you can see that people believe that the federal government should assign a higher priority to improving STEM education. But there's another way to make a difference, and that's for each and every one of us to practice a little bit and start using a very simple message frame for communicating about what you do. It's called aspirational communications, using the then, now, and imagine framework. We use it here for sickle cell disease, but it can be used for anything you're working on. Involves connecting with people in terms of what they might remember from previously when a particular disease or disability could have been a devastating diagnosis to hear, or even a death sentence. But today, thanks to long-time public and private investment in research, we're at the point where we have new treatment options, let's say. But the real aspirational, exciting goal is the cure. Imagine that world. So being able to talk to people who aren't science trained in ways that meet their needs and expectations, we know works, and we know that the ASG, by the way, is masterful in creating these then, now, and imagine frames. So I encourage you to practice amongst yourselves, learn more from ASHG, and use these in new ways with people who you've known a long time, who've never really understood what you do and may ask you puzzled questions about it from time to time, or to strangers whom you meet for the first time, including strangers like perhaps your new member of Congress when that person takes office. And as a longtime advocate, I think boiling it down, the four most important words that any scientist can say and convey in order to help make the case for more science, for more possibilities, for more solutions to be found, is when somebody asks you what you do, instead of saying that you're a geneticist or a microbiologist or even a scientist, say, I work for you. I work for you, because you do. You serve the public's interest, and trust me, the public wants you to succeed now more than ever before. Thank you very much.
Now that we can go to our discussion. So everybody can hear me? Okay, so that was really terrific, and I really thank our four speakers for really illustrating the growing tent for genetics and genomics in a number of different domains, both going on at NIH, the private sector, and then thinking about it through a lens of advocacy. So we have a number of questions that are coming in from various attendees of this virtual meeting. And I'm going to try to, working with Tony Winchell-Boris, we will try to pick some of these questions. We won't get to all of them. I'm going to start with George, because I know George might get pulled away and may not be able to stay to the end of this session. We certainly have a number of questions coming in. Maybe I'll start with one of my own. George, you gave an example, and since this is a genetics community where sort of genetics, sort of some of the earliest genetic studies with COVID-19 actually ruled out a pathway that otherwise could have been a potentially very interesting target. I wonder if you have any early clues from any of the initial data of possible targets or possible pathways that look promising. and whether you could talk about them at all, or at least acknowledge that there's some developments coming down the pipeline.
Yeah, no, I think that, and it's not only coming from our labs, but certainly pathways that are known to be involved in the response to viral infections, particularly the interferon pathway and others. There's also a number of target genes that are associated with UAS locations that have not yet been definitively narrowed down to individual target genes, but where there are, as we all know, a lot of potential genes located within that area, several of which could be candidates. And I'm sure not only us, but many others are following them along. However, as I was getting to, in some cases, when you have an exogenous target, such as the virus itself, that might represent the most obvious direct target. Just block the direct virus, as we're doing with our
So I'm going to wrap up the questions and pick George, making them a little bit harder each time. But one of the attendees typed in they were interested to hear sort of, and especially in light of some of the earlier speakers hearing from about inequities and disparities. And what is Regeneron's sort of current thinking as it starts to roll out COVID-19 treatments to sort of make them accessible, affordable, available to everybody in a growing tent, and with that kind of a theme? And I'd just be curious what the current corporate thinking is around this public health emergency.
Well, one thing that I think is important to know is that groups that are generally underrepresented in clinical trials, probably because of their overrepresentation in this horrific pandemic, were actually well represented in our studies. So the results that we're actually seeing, hopefully we will know that they do pertain in those populations as well. I agree, it's a very difficult issue in terms of making a limited amount of drug available. As you know, these biologics, particularly these antibodies and antibody cocktails, you can't easily make millions and millions of doses. We initially started to manufacture at risk. We set off on this path back in January and February before we all knew how horrific this pandemic would be, and we dedicated our entire North American facility at risk, at our entire risk, just in case it would be a horrific pandemic and just in case we could come up with a therapeutic. Even with that dedication, right now, with that entire plant dedicated, we can only make about 100,000 doses a month. Luckily, we engaged Roche Genentech as a partner. They can hopefully by the beginning of the year be doubling or tripling or quadrupling our own internal effort. But luckily also the Department of Defense came in, and while we started the process and took on the costs entirely at our risk initially, they came in and they decided to cover the cost for the first 300,000 doses. And it's going to be their responsibility. It's going to be the government, and it's going to be up to our represented officials to determine how these first 300 doses will be distributed and to whom. And of course, we all hope, we know these are going to be very challenging and difficult questions. They have committed to making them available free of charge to whomever they make them available to. But obviously, these are going to be limiting reagents. Even if both our cocktail and the Lilly approach get authorization in the near future, this will still be a crying need as we're seeing the epidemic proportions that we're now facing actually growing at this time. So who is going to get a limiting drug is going to continue to be a challenge for a while, which is why we also need advances in the near term of vaccines and so forth to try to reduce this crushing need. But that's something that I don't think is going to be an easy question to answer.
So let me shift from a more social question to now back to a scientific question. And actually, there's two I'm going to sort of combine, and you could answer each half. One question that came in is sort of, what do you see as some of the genetic and genomic tools that are really needed that would help advance development of therapeutics along the lines of what Regeneron is interested in? So that's just sort of at a technical level, what's high on your wish list? And then a corollary of that, which I was very curious, is here you have on this panel including me, 3 directors of NIH institutes. And I guess I'd be curious, because I know Regeneron's very involved in a number of consortium and a number of partnerships, but you're in part of the ecosystem that has a different view, perhaps, of NIH. And here, with 3 institute directors on this panel, what guidance, what advice, what suggestions would you give us to that would sort of strengthen the ability, in particular, to use genomics approaches to advance the kinds of things that Regeneron is attempting to do?
Yeah, well, and I know I've said it, Aris Barris said it, many of us have said it, that size really matters. We have started to make discoveries based on rare sequence variants that you could not pick up with GWAS and chip approaches and so forth. You can only pick them up by sequencing literally millions of people. And we're now seeing that these exist. And so we really do think that, especially when you're talking about rare diseases or diseases where you're looking for rare responses and so forth, it would be great to have as large a database as possible. We have, as you know, Eric, we are so willing to work because we consider most of this pre-competitive. That's why we started the consortium to sequence the entire UK biobank and raise the funds in order to do it and made all the information publicly available. We think that the key to these large efforts is really having the right collaboration and public-private partnerships. We would love to work as closely and as powerfully as we're working with the National Health Service and the UK Biobank overseas, right here in the United States with the NIH. We think that we should have started these efforts years ago, but we think the power of putting the different things that people can bring to the table together for these large pre-competitive efforts can make a huge difference, because our dream is to basically have what we all would love to be able to use to answer immediate questions. If we had a national database that basically had everybody sequenced and had everybody's medical records, everybody's response to every drug that they're taking, for all we know, there's some existing drugs, not IL-6 blockers, but other existing drugs that are out there that might have impact to prevent, to treat, that we could be realizing from these databases and putting together the genetics and putting it all together. And so I think that that's a shame to me that we haven't figured out as a country how to actually create what could be one of the most useful databases ever. So I think that should be our goal. Sequence everybody, get everybody's medical data ongoing on a real-time basis, so we can make all the associations with genes, with treatments, with every level of intervention and care, to really understand in real time what's working, what's not. And so we don't have to wait. We don't have to wait, as we did 3 months after our clinical trial, to find out that genetics said that IL-6 is not a good target for COVID-19. And if these things had been available sooner, if they're available now, and I think we as a society and we as a country should be working together to figure out how to do that.
Thanks. That's really helpful. Let me shift gears and go to Mary Woolley. Mary, there are a number of things that are coming in and that also that I want to ask. Let me start with one, since this is a genetics audience. And I thought it was great that you showed some data about people's thinking around what human genetics is all about. I guess one of the first questions I had is, how does that compare to other areas of basic translational and clinical research?
Well, Eric, I'd say that people are immensely interested in and supportive of research across the board, whatever topic you pick. If you make it too hard to even understand what the question is that you're asking them, they're, needless to say, not going to be as interested in it. But if you can connect some dots to what's it got to do with me, how's it going to benefit me and my family, people will say yes, they're interested. And the thing they're most interested in typically is getting the solution, getting the answer. I want to say something about George's important challenge just now, that I actually think we can do as a nation. It'll take a major advocacy effort, but this is the perfect time to do it, to challenge our fellow citizens, first of all, and then our policymakers to respond, to assure that everybody is involved in research all, as a matter of course, all the time. Most of the time passively, by putting their data to work, so that it's all in for everybody. Yes, of course you could opt out. Of course you can opt out. But most people, I believe, are going to opt in. The privacy concerns are nowhere near as high as some people believe. That's been tested through various surveys. And we should take heart in things now that we take for granted, like donating organs that you put on your driver's license. Before that was there, it wasn't there. Now it is there. Now people consciously make that decision to help others. And participating in research as a normative value that's reinforced by the healthcare professional community, I think it's an achievable goal. And I'd love to talk to you about that sometime, George.
So Mary, this actually overlaps a question that came in from someone and I'd be curious to learn a little bit about how you handled this. So one of the questions that sort of came in was, how do you deal with issues like access to genetic and genomic testing in clinical settings when it starts to relate to issues about accessibility and relates to disparities and inaccessibility? And as you just pointed out, if everybody who was getting clinical care would be participating in research, it would be a great enhancement to the entire enterprise. So as an advocacy group for research, do you sort of put a boundary between advocating for science and then getting into the very tricky waters of universal health care and Affordable Care Act and all the things going on here in the United States? Or do you stay away from that? Or are you willing to jump into that? Because clearly these things start to become interrelated, especially when you start dealing with the science of health disparities.
Right. Well, my answer is kind of complicated, Eric, because our society is complicated right now. But the big breakthroughs that have been driven by advocacy all started out small, all had to face tough challenges. If you think about the smoking challenge, the tobacco challenge, or assuring that women could participate in clinical trials, or the HIV/AIDS activists demanding that science and regulation move faster. A lot of things, a lot more is possible than we sometimes think meets the eye. And will it, melt over into some currently very, very contentious issues? Of course, that's going to happen. And we would need, frankly, for a campaign like I was just talking about with such enthusiasm, we'd need a lot of partners. But I know there are people out there who are interested in this. So to hear George describe is great.
Tony, you wanted to interject something?
I did. I wanted to just follow up with Gary and Diana from the NIH perspective, and then George, because he's generating large data sets. What are the barriers that our society can help you with in terms of making sure that we have such a diverse participation, and not only in research, but obviously engaging our communities? And also, Mary, of course, you can pick this up as well. But the most important thing is, what are the barriers that you guys see at the NIH level or at Regeneron's level? That would be great, I think.
I guess from where I sit, I guess that commitment to inclusion. I hope that the 1.5 million genomes that Regeneron has with Marshfield, et cetera, is reflective of the diversity of our country. I'm concerned that historically genomic resources have been incredibly Eurocentric, and we just keep chasing our tail and not making progress there. So I'd love that if we're going to make a big tent, make it an inclusive tent, and one in which we're very intentional about that aspect of it. Who we phenotype, who are in these big data collections, and recognize that they're just not going to raise their hand and volunteer. That takes outreach, that takes engagement, that takes partnership, and that takes them being at the table. I hope they're at the table. I guess I don't know if that's why I'm on this panel, but I don't know how often it's an inclusive conversation to be sure that they're part of this discovery as well as the benefits of the testing or technology. This notion that we don't want to get into a social sphere, I think is, quite frankly, naive. You can't address these things that are structural and systemic and remain pristine within an ivory tower. And that's going to be uncomfortable, but that's what needs to be done. Thank you.
Yeah, so I could add to that. I think one of the biggest barriers is a lack of trust. Unfortunately, I mean, you can go from Henrietta Lacks either backwards or forwards, but there are many examples of, how research did not include the participant. We often say no research about me without me. And that includes all of the communities that we advocate for at NICHD. I mean, particularly maternal mortality. We need to encourage participation of the women who are at the highest risk, but there are so many structural factors that have to be dealt with in addition to just the research aspect. So we're operating within a much larger societal issue, but fundamentally we have to establish trusting relationships. I think we have to do a better job at communicating the benefits to different populations of research. And I totally agree with Gary. We need to include the populations. We need their input. What I think from my position of white privilege, may not be relevant to certain populations and likely isn't. So we need to have people at the table. I do want to tie together two things that Mary said and George said. So imagine if we had a newborn genomic sequence at birth, because babies that are born in hospitals or clinics that have midwife services do get newborn screening. And, I'd like to imagine a future where you have your genome sequence at birth and that can inform not only the potential future health of that child, but also that information can be anonymized, but also linked with electronic records to facilitate research. So, I think that is the ultimate goal, at least from just purely my opinion.
Yeah, maybe I can add to echo some of these comments, and certainly what Gary was saying is this is what we certainly see. I mean, in the COVID trials, as I said, because of the demographics, usually underrepresented populations were there. But for a lot of our genetics efforts and so forth, I think it's going to require a lot more outreach. And we are really thinking very hard about the right ways to do it, because I think it makes sense not only from the representation side in terms of the fairness argument, but also the scientific side. To get diversity in there will lead us to more information and ultimately more answers for everybody. But I also want to get back. I thought Mary's talk was great. I think that it's about changing attitudes. I really do think that we, I'm a child of the 60s. I'm a child of, JFK's dreams and moon missions. And I think that this is what we need. We need great leadership to really change the way we're thinking. And we need to elevate science in the, public consciousness. And we need to make scientists heroes. And the scientists who are heroes can't be the ones who are on the lists that that Mary had there. And I think that we really have to go after really big, bold efforts. I think, Eric, you asked it. I think that we should all get together and we should do it. We should be doing a 10 times better job than what the United Kingdom is doing. We should have a really serious mega scale national effort to make the sort of databases that are inclusive and so widespread that they will be providing prospective information that will take science to that whole next level, that'll be an engine for real innovation and change, because we see the power of what we have right now. And if we can increase the diversity in the numbers by orders of magnitude, it can make an enormous difference. And so I think we need leadership that can think big and drive really big efforts and really big change.
So George, you just picked up on something that one of the questions that came in. I want to go back to Mary Woolley, and which really you are a child of the 60s, similar to myself. Mary, Research America has been doing surveys for a long time of different groups, of different populations. Is there evidence that the different generations, especially younger folks, are going to bring a different perspective and a different prioritization that may influence participation in research, may influence respect of science, et cetera, et cetera? Should we be optimistic based on sort of the millennials, the Generation Zs, and so forth?
Yeah, good question. First of all, let me say that I'm a child of the 60s also. Shook JFK's hand, out of that. But the question you just raised right now, Eric, and public opinion surveys is only one of the things that Research America does. So we're not survey experts per se. But I've talked to a lot of those folks over time, and one of the questions that really perplexes me right now is in this most recent survey that we commissioned, young people, that 18 to 29-year-old cohort, tends to be generally less positive / optimistic about the future period, and science-driven future specifically. Tends to be more negative about institutions, although in general our society is more and more negative about institutions. We don't really understand what's going on with that young cohort. They're not children of the 60s. They're children of some horrific things that have happened in their lifetime, not going to the moon, but rather 9/11, incessant wars, a recession, now COVID, a lot of pretty brutal things. Not that our generation didn't have brutal things as well. We did. We had Vietnam, for example. And we had racial tensions that still haven't been resolved. So I don't understand. I wish I did. We're going to take a harder look. I think it'll be mostly by asking other people to share information with us about that younger cohort. And I think we will also find out after the election, not only whether they voted, but how and for whom. And that's going to tell us some stories too.
Tony, you wanted to ask something?
I only wanted to give you a chance to give your opinion about the question that we heard everybody else respond to, which is how to increase the diversity of the populations that are being sampled. And given your position, I thought you might want to say something as well.
Well, it's something that's an incredibly high priority for NHGRI. If you read our new strategic vision, which was published today, actually, in Nature, we in multiple places make the same strong plea that this has got to change, that we as a community have got to to do better when it comes to diversity in participants, diversity in our workforce. It's exactly what Gary Gibbons was saying. We've got to have, it's not just the participants, it has to be the people coming to the table and doing the science and engaging with these participants. I could certainly say that our major programs, and Terry Manolio, who's this year's program director for this entire meeting, is an incredibly strong advocate and a very effective one at the Institute as one of my division directors, and in many of the things under her guidance in terms of our extramural program have relentlessly put into these new programs a very high bar for what is expected in terms of diversity recruitment, including special programs that are particularly focused on diversity recruitment. So, NHGRI, we're a small institute, but we are absolutely doing our part, and we are strong advocates for this improvement in the scientific enterprise. And I guess with that, I want to pivot back. The question I wanted to turn to Gary and Diana as my two institute colleagues on this panel to ask is and George sort of put the call out to us, and we have the All of Us research program, and that's clearly a signature program for the Institute and is doing quite well in its recruitment of traditionally underrepresented participants. But, what should we be doing more? I mean, Gary, in particular, I know your institute's doing a lot of things. Diana, you are as well. But is there sort of a time for us to take a step back and even think of different approaches? Maybe structurally, the way we are is sort of inhibiting our ability to do better. Are there things we're not doing that we should be doing as individual institutes? And I know that's a hard question, but I'm curious if either of you have any immediate thoughts.
Well, I would say so. That. You want to go ahead, Gary? Or
no, go ahead, Dan. Go ahead.
I was going to say that I do think that we're going to have an incredible opportunity when we see the end of COVID. I mean, this has been a global pandemic. It has brought the world's attention to science, and the solutions will come from science. I love Tony Fauci's quote that pandemics shape societies, and there will be societal change. And I hope that one of the things will be that there will be increased public interest in science, particularly if there's a vaccine or a treatment that, we know that we've already made progress since the early part of the year. But, can we leverage that opportunity writ large for science? And then, how do we apply that to our individual institutes? So we're particularly focused on the fact that pregnant women get much sicker with COVID than women of the equivalent age. And again, there's health disparity there. Latinx and African-American women are more severely affected. And we know that is because there are other underlying predisposing risk factors. So we are certainly reaching out to use this as an opportunity to include, a much higher percentage of underrepresented populations in the work that we're doing that is focused on our particular response to COVID. But our strategic plan, I didn't have time to go through all of it, but addressing health disparity, health inequities, that's woven through our strategic plan. And, we deal with the future of America, and the future of America is not white. And so we want to make sure that children of any color have the same opportunities to live a healthy life.
Gary?
I would echo and underscore that. Maybe it goes back to Dr. Yiannopoulos' point about the GWAS after the drug. So similarly, where were we before the pandemic in these communities? We knew the health inequities that they were already experienced that were laid bare by the virus. And so I hope that what we gain out of this crisis, as was said, that science serves, that science solves problems, science saves lives, but we have to prove it in the most vulnerable, whether it's children or these communities of color. That to me will be the testament of whether science is doing what it's supposed to do for the public good. As Mary said, we work for you. This is our time to prove it and put up put our actions behind the rhetoric, and really build in that capability, the resilience for the next challenge. So I hope that's what we do and redouble it. This won't disappear after the virus goes away. These things will still be there. That's what we have to solve. I hope that the resource we create that is able to anticipate their risk and their response to treatments, as was said, extends to the whole human family. And we already know scientifically, we'll learn actually more by studying the course and trajectories of these communities than almost any other community in our country. That's where we have to get better. And we haven't been good enough. Over.
Well, Tony, I'm going to turn this back to you. I think this has been a terrific presidential symposium. I hope you've been pleased with it. I certainly enjoyed participating in it. I think the four speakers have really brought up a number of incredibly interesting, important, and take-home messages that I think give us a lot to think about.
Yes, well, definitely. Thank you, Eric, for co-moderating. Thank you for all the speakers, panelists, for all of your wonderful insights. And thanks to all of the audience for participating. Have a good evening and enjoy the rest of the meeting.
Thank you.
Thank you.